Dear students, this is our 21st lecture on medicinal chemistry and 7th lecture on antiviral agents. In the previous lecture, we have discussed about chemistry of COVID-19, and I have told you about some drugs which are used for the inhibition of COVID-19. So today in this lecture we will discuss about synthesis of some of the commonly used drugs against COVID-19. And this I have already covered in the first lecture that how the first known case was identified in Wuhan, China, wherein we came to know about this disease, which ultimately become one of the few most dreaded diseases in the form of pandemic. And these types of pandemics, they are generally caused because of some social and environmental factors, including climate change, or when we play with the natural ecosystem. The destruction of this natural ecosystem also affects and bring about these types of diseases, which ultimately are converted into pandemic. The effect of this disease, COVID-19 pandemic, was so quickly spread that by this year 2024 July, um, as per the record given by WHO, more than 7 million people have already died due to this pandemic. So, such was the impact of this pandemic on mankind. So, this was the first lecture wherein we have discussed about the structure and how the action of these viruses take place in our body, the mechanism of action, and the whole replication process of this virus. And in the previous lecture only, I have discussed about that how some of the already known drugs which have been developed for some other antiviral or other activities, which are having some specific activity against some other diseases. They were utilized for inhibition of various steps of replication process in case of COVID-19. And now in this lecture we will discuss the synthesis of some of these commonly used drugs for COVID-19 disease. And as I have told you during the replication process, that drug development will be based on the possible binding sites between human proteins and COVID-19. They have been discovered. Uh, this binding site means when the entry of this COVID virus will take place in the human cell. So, if we know the uh, complete mechanism of this binding process, that means we can definitely tell that which type of compounds or drug molecules they can be used which can act as inhibitors so that this virus cannot enter the human cells. So, host binding and subsequent fusion of the viral and the host cell membranes, they are basically caused by one subunit of the S proteins in case of SARS-CoV-2. And binding of the S proteins S1 subunit take place to the ACE2 receptor in the human cell, and then the computer simulations, they were utilized for uh, the chemistry of the attachment of this for developing a novel disease for curing the COVID-19. And on the basis of this simulation, shape of the virus was also determined. So basically, drugs can only be developed if we completely understood the chemistry of the replication process and the mechanism through which the various steps in the replication process of the virus take place. So only then we can identify the drug molecules which can be used to inhibit those activities in the replication process. Uh, at present, there are not any committed medication that can treat COVID-19 completely. But a number of well-known antiviral medications which have been used for a long time to treat AIDS, MERS, SARS, malaria, and other infections. They are being investigated in clinical trials in relation to COVID-19 also, so that they can be useful against SARS-CoV-2 virus. Now, the most promising repurposed medications which have been used for COVID-19, they are fusion inhibitors, protease inhibitors, nucleoside reverse transcriptase inhibitor, nucleotide reverse transcriptase inhibitor, and neuroimmunities, which are inhibiting the release of the virus from the cell to the body. So most commonly used fusion inhibitor is arbidol, and basically this arbidol is used against influenza. So this is an anti-influenza drug, and its use for COVID-19 has been approved in Singapore and China. Then protease inhibitor, there are plenty of protease inhibitors which have been used, and most of these you can see they have activity against HIV. So they are anti-HIV drugs which have been used as protease inhibitor in case of COVID-19. So lopinavir is used in US, Japan, Singapore, Italy, and then the mixture of lopinavir and ritonavir has also been used. Then atazanavir has been used in Singapore, darunavir in Italy. Here also lopinavir, ritonavir mixture as a fixed dose has been used. Then secunavir has been used in Singapore. Similarly, nucleoside reverse transcriptase inhibitors like amtristabine, which is an anti-HIV drug, is used in Singapore. Then nucleotide reverse transcriptase inhibitor like remdesivir, which is used against Ebola. This has been used in Singapore, US, IPC, WHO, and Italy. Then neuroamidase, that is viral release inhibitor, ostelmavir, that is Tamiflu, which is anti-influenza A and B drug. This has been used in Singapore, IPC, and Indonesia. So these are the most commonly used antiviral drugs which have been found effective for use against COVID-19 also. So first is hydroxychloroquine. Chloroquine or HCQ. This is an anti-malarial drug. Uh, this has also been used for the treatment against COVID-19. Uh, this has, as I told you, that this has shown anti-malarial activity. Uh, And in malaria, it prevents heme polymerase in malarial trophozoites from doing its job, which kills the parasite. Whereas this is also a weak organic base because of the presence of these tertiary, secondary, and tertiary amine groups in the molecule. And because of this weak organic base nature, this passively diffuses across cell membranes for SARS-CoV-2. And when it becomes protonated, it increases the pH, and because of increase in pH, it prevents virus particles from fusing and entering the cell. So it can act as an inhibitor to entry and fusion in the virus replication cycle. And further, it was discovered that combination of this hydroxychloroquine and azithromycin, this was more successful as inhibitors of entry and fusion than using HCQ alone. So this is the structure of this drug. Basically, this is a quinoline derivative, and its synthesis was first reported in 1950-51. Uh, there are three major steps involved in the synthesis of HCQ. First step is alkylation of N-ethyl N-hydroxyethylamine. So this is N-ethyl N-hydroxyethylamine. When this is reacted with 4-chloropentane 2-one, this one in the presence of 1,2-dihydroxyethane, in presence of paratoluene sulfonic acid in cyclohexane at 80 degrees centigrade, uh, the nucleophilic substitution of this attacking at this place, attacking at this carbon of the carbonyl, then followed by cyclization at this place, will lead to the formation of this cyclic 1,3-dioxolone derivative. And this 1,3-dioxolane derivative, when reacted with one more molecule of 2-ethoxyaminoethanol, this is converted into uh, this molecule, where uh, the replacement of this part take place with this amino alcohol. That this can be converted into this tertiary amine by alkylation process, an alkylation process, which can take place either directly on reaction with xylene and NaCl, it is converted into this, or through this process we can synthesize C. Uh, Whereas this method gives us more improved yield of the order of 67%, whereas if we do it directly, only 44% of the product is formed. Then this tertiary amine C, uh, when this is reacted with hydrogen in ammonia in presence of pyronickel reduction, first hydrogenation followed by amination will lead to the formation of this type of amine, and this amine is formed by reductive amination of this tertiary amine. And in the final step, this amine reacts with 4,7 dichloroquinoline, and the process of this amino group will get attached to this carbon, and we will get HCQ as the product. So this is how hydroxychloroquine can be synthesized from 2-ethoxyaminoethane 1-ol. Then another improved method was given in 2018, where uh, high yield continuous flow synthesis of hydroxychloroquine was achieved, and this methodology is different from the original census in two ways. First is that here SN2 displacement was made easier by switching the chloro group which was present in the first case in the ketone to ido group. So instead of there we have used chloro group, but here 5 ido pentane 2-one is used, and this can be synthesized from 3-acetoxyoxylane 2-one on reaction with hydroiodic acid, wherein decarboxylation of this will take place, and we will get 4 ido, this is ido not chloro, 4 ido pentane 2-one, and then this 4 ido pentane 2-one will react with this ethanolamine, substituted ethanolamine in the same way as we have seen in the previous case, and we will get this type of tertiary amine which was synthesized in the previous case also. And the second difference is that here a two-step process involves the synthesis of an oxime. There we have in a single step converted this tertiary amine into this primary amine through reductive amination. But in the present methodology, what we have done is first we have synthesized oxime of this ketone like this. So we know that nucleophilic addition of hydroxylamine to carbonyl compounds can lead to the formation of oxime. So this oxime can be synthesized very easily, and then this oxime undergoes reduction in presence of rhine, nickel, and hydrogen in 84% yield to give us this amine, and then this amine is condensed with 4,7-dichloroquinoline in the same way to give the product, that is HCQ is produced in 78% yield. So this method A has been found to be more effective and has given more yield in comparison to the previous method. The next drug is remdesivir. Remdesivir is used for the treatment of Ebola, and this has been found to be the most promising drug for COVID-19 because of its wide range in vitro activity against coronavirus, which includes SARS-CoV-2 also. And this has been authorized by US government for emergency used in fighting COVID-19. This is basically a monophosphate nucleotide analog prodrug, that is a compound after administration that is metabolized and converted into an active drug. So three fragments of this drug, they are nucleobase adenine unit, then pentose sugar unit, and then phosphoramidate unit, and all these three units they are responsible for different activities of this drug. So these retrosynthesis of remdesivir shows that this might have been formed from say hydrolysis of this bond will lead to the formation of this adenine ribose nucleus part and phosphorus part, and then this on breaking at this point will lead to ribose and adenine part. So combination of ribose and this adenine part will lead to the formation of this part, and then this phosphate ester on reaction with this will lead to the formation of remdesivir. So this is the disconnection approach, which tells us about the possible uh, starting materials for the synthesis of this drug. The advanced synthesis of remdesivir is basically achieved by the coupling of phosphoramidate ester group, this one, with the nucleoside part. So both these they will combine to give us remdesivir as the product. And now we will independently first synthesize this part, then this part, and then both of these they will combine to give you remdesivir. So first is synthesis of phosphoramidate A. So phosphoramidate A is synthesized from chloridate racemic mixture. This one, which was in turn synthesis by alanate ester. This is synthesized from the corresponding amino acid. This alanate ester on treatment with phenyl phosphorodichlorochloridate in presence of triethylamine and dichloromethane, uh, along with the 4-nitrophenol, will lead to the formation of this racemic chloridate. And this chloridate on in presence of isopropyl diisopropyl ether, uh, this gives us the preferred diastereomer A, which is this is a racemic mixture, and then this can be converted into our preferred phosphoramidate diastereomer A, and then this A will be condensed with the B. The nucleoside B is synthesized from this benzyl protected ribolactone, which is synthesized from the corresponding ribose sugar. So when this substituted ribose sugar undergoes oxidation of this hydroxyl group in presence of acetic anhydride DMSO, this gives us this benzyl protected ribolactone 1, and this reacts with iodonucleobase, and this will add to the this carbon oxygen bond to give us C nucleoside analog 3, and this is C analog C nucleoside analog 3 then reacted with TMSCN, TFOH, then TMSOTF in presence of DCM at 0 degree centigrade. Basically, the hydroxyl group is converted into cyano group in this case. So this cyano group was introduced by using TMS-SCN and triplet trimethyl triplet and trifluoroacetic acid to produce this nucleoside derivative. Then this nucleoside derivative undergoes hydrolysis of these benzyl groups in presence of BCl3. So benzyl deprotection was carried out by BCl3 to give us this nucleoside B. Then this nucleoside B interacted with the second part, phosphor formidate A, which we have synthesized. The coupling of these two, followed by treatment with HCl, led to the formation of prodrug remdesivir. And as you have seen, that remdesivir can be synthesized from two different parts, A and B, phosphoramidate and nucleoside, which couple to give us this drug. The next is the combination of lopinavir and ritonavir, which is used for COVID-19, and this combination was used against HIV-1 protease inhibitors, and this was effective against SARS-CoV-1 also. So we will discuss the synthesis of both of these drugs one by one, but in this lecture we will discuss the synthesis of lopinavir. The synthesis of lopinavir was mainly done by Abbott Laboratories, and retrosynthesis of this drug suggests that three fragments they are required. One is this amino alcohol 2, and then these side chain carboxylic acids. So one is this is carboxylic acid, other is this carboxylic acid, and these two amide linkages, they are to be broken in the retrosynthesis to have all these three fragments which are used to synthesize this drug lopinavir. Now first the side chain 3 is synthesized. This 3 is synthesized from amino acid alvaline. So when alvaline is reacted with lithium chloride in lithium hydroxide with this acid chloride, phenoxy acid chloride, then basically this will react with this amino functional group at this place, and we will get this type of product. And then this N-terminus carbonate uh, in the presence of sodium hydroxide is converted into C when this reacted with this amino chloride. Again, here the same thing. The amino group will attack as a nucleophile to this carbonyl system with the elimination of phenol derivative, and we will get this type of product. And then this product will undergo cyclization at this nitrogen will attack at this position with the elimination of this halogen in presence of strong base like tertiary oxide. So this side chain carboxylic acid, which is one of the units in the synthesis of lopinavir, is synthesized. Then central core amino alcohol is synthesized from L-phenylalanine. So when L-phenylalanine is reacted with benzyl chloride in presence of uh, potassium hydroxide and potassium carbonate as bases, then benzylation of this nitrogen twice and this oxygen one. So basically we can say that tribenzylation of this alphenylalanine take place to give us this tribenzyl ester as the product. And then this tribenzyl ester reacts with acetonitrile A9 generated from this generated from the reaction with sodamide. So sodamide is a strong base. This will generate a carbanion at this methyl, and that carbanion will attack at this carbonyl, and this will be eliminated. We will get this type of cyanomethyl ketone as the product. And then this cyanomethyl ketone is converted into enamine when this is reacted with benzyl magnesium chloride, and the reaction is highly stereospecific with more than 99% of enantiomeric success. Then diastereoselective reduction of this enamine take place, and this will be this sodium borohydride reduction will lead selectively reduction of carbon-carbon double bond will take place, and carbonyl functional group is not reduced. So this will lead to the formation of this amine. Then further reduction of this amine in presence of uh, tri ethanol triethanolamine leads to the selective reduction of this uh, into a stereo specific product, which is our central core amino alcohol, formed in 89 to 93%. So this is the second part of the drug which have been synthesized. First we have synthesized the third uh, part, that was acid part, now amino alcohol part, and then this uh, the first carboxylic acid part which we have synthesized. Then this amino alcohol, then and the acid chloride which is synthesized from the acid which we have synthesized. When this is reacted with iron chloride, the corresponding acid chloride will be formed. Then this acid chloride will react with the central core amino alcohol which we have synthesized in the previous step, in the presence of imidazole as the base, and this will be converted into this type of amide. So amino group will get attached to this carbon with the elimination of Cl from here, H from here. We will get this type of amide which will be formed. Then this amide will be converted into the deprotection of this nitrogen from these benzyl groups. So debenzylation or deprotection of these benzyl groups will take place to release the primary amino. So this B will be converted into this amino alcohol C, and diastereoselectivity of this amine was improved by crystallizing as an amine PGA salt, and this amine PGA salt then have the purity of 98.5%, and in the final step this amine PGA salt will react with the side chain carboxylic acid to synthesize the lopinol. So acid will react with this amine to synthesize the second amide linkage present in the longevity. One amide linkage is this one. Other amide linkage will be synthesized when this amino group reacts with this carboxylic acid to synthesize the. So this is how lopinavir is synthesized by coupling of the three subunits which we have prepared in the previous slides. So these are the references which have been used for preparing this lecture. Thank you very much.